I will.
Sorry.
So I'm going to give my welcome. So I believe this is the sixth GREG seminar. I think we still have at least two to go. So first I want to introduce myself. I'm Anne Charmentier and I'm president-elect for SSE. And well, I wanted to give a little speech about what the GREG excellent grants are about, but maybe I will just post on the chat the link to the SSE GREG Award if Gavin wants to learn about them and doesn't have the details yet. And I will directly introduce today's speaker, Claire Williams. So Claire is a PhD candidate at the University of Nevada in Reno, and she's working in the group of Michael Logan. Her work is focused on how environmental change affects the gut microbiomes of animals. And she has investigated this question in an area of host species. She is just back from the Smithsonian Tropical Institute, a research institute, where she studies tropical anolis lizards. It sounds like a very long but exciting fieldwork. And today she's going to tell us about how microbial communities can alter host phenotypes in important ways. And she's probably also going to argue that understanding how hosts respond to selection requires understanding how host-associated microbial communities respond as well. So Claire will give her seminar and then we can have a session of question until the hour. And even though it's not many of us in Zoom today, I still want to remind you that we expect everyone to follow the code of conduct for any question or interaction that we have. And we're also going to place the link to the code of conduct in the Zoom chat. Claire, I am very eager to hear about your research, and the floor is yours.
Thank you. Okay, I'm just gonna share this. Okay, you can see the screen okay? Okay.
Yeah, perfect.
All right. So thank you for that welcome. Again, my name is Claire. I'm a PhD candidate at the University of Nevada in Reno. And I also have worked at the San Diego Zoo Wildlife Alliance and the Smithsonian Tropical Research Institute during my master's and my now PhD. I did my master's at Trinity College Dublin in Dublin, Ireland. And so kind of throughout the last, let's say 3 to 5 years, I've been really interested in how the gut microbiome can mediate host responses to environmental change. And I'll tell you a little bit about what I mean by that. And I'm going to tell you about a few different studies that get at different aspects of this topic and this sort of question. So just to give you a little bit of background on kind of why I got interested in this. I started out in undergrad really interested in microbes themselves, kind of basic microbiology, culturing bacteria from soil, seeing what types of metabolic pathways they have encoded in their genomes, testing whether or not they produce antibiotics, that sort of thing. And I was really interested and fascinated by how unique and diverse and beautiful these microbes were, growing them in culture and seeing all the different colors and the colonies, and just thinking about how prevalent they are all around us, and we rarely ever really consider or think about them except for maybe when we get sick. And then kind of later on in undergrad, I read this paper in one of these classes that I was in that started to talk about how the gut microbiome in humans and in mice could affect kind of depression and mood, because microbes in the gut were starting to be shown to produce and modulate neurotransmitters, and those could actually make their way up to the brain where they actually affect the way that we feel. And I found that really fascinating because, the way that we think about ourselves and how we feel and what we think, that feels very human, very personal. It doesn't feel like something that could be, easily manipulated by things that we don't even, invisible microbes. But it seems that that's going on, and that was really fascinating to me. And so then eventually I found myself at the Smithsonian Tropical Research Institute, where I started to investigate how gut bacteria could potentially affect thermal tolerance in lizards. And so that kind of tied together these interests for me, thinking about how environments were changing, how gut bacteria could affect something that might be super important for environmental change, like thermal tolerance, and thinking about how all those things tie together to affect how animals respond and survive under rapid environmental change. So as I kind of mentioned, I started really thinking about animals as meta-organisms. So not only as their own genomes are made up of their own genomes, but also having access to a wide array of genetic diversity that's encoded in bacterial genomes in the gut and in other areas of the body where microbes reside. I'm mostly interested in the gut microbiome just because in my study species, it seems to be maybe where the most important interactions are between host and microbe. And I primarily focus on bacteria. So that's what I'm going to be talking about today. And so if you think about the host and microbes as interacting kind of as one large organism, you can think about how having a microbiome might change the phenotypic landscape in a population. So you could see this dashed line here maybe is the variation in a phenotype without any microbe contributions. And then with the contributions of the microbiome could actually shift the mean or standard deviation or both of that trait. And so just some examples of that are this study that was done in wood frogs where there was kind of a cross transplantation of microbes. And so wood frogs were inoculated with bullfrog microbes, and it dramatically shortened their time to metamorphosis. And so you can see that this solid line is kind of the wood frog inoculated with wood frog. It's like the control. And the days to metamorphosis is much earlier when they're inoculated with different microbes. And in this way, you can see they're accessing phenotypic space that didn't really exist before. And the same thing has been shown here in Drosophila, showing that body length can be modulated by whether or not the microbiome has been disrupted with antibiotics or not. And so these are just kind of two examples of how the microbiome can affect host phenotypes. And so some work has been done trying to kind of integrate these ideas into standard quantitative genetics. Kind of previously, we thought about phenotypic variation being made up of host genetic derived variation and environmentally derived variation. But we can maybe start to think about how variation in the gut microbiome can also contribute some amount of phenotypic variation. And if we see variation in phenotypes, then that could scale up to alter host fitness, especially if those phenotypes are under selection in some particular environment. And so a kind of straightforward example of this is some work that my master's thesis advisor did, and I worked a little bit on this project. And effectively, these were captive rhinos in the San Diego Zoo. And one of the main problems that they were facing was really dramatic infertility in captivity. So they brought these southern white rhinoceroses into captivity in order to breed them, to have a stock kind of population, because their numbers were dwindling due to poaching, but found that they were having a really difficult time getting these rhinos to actually successfully reproduce. And through some investigation, they found that part of the issue was that the diets that they were feeding the rhinos were very high in phytoestrogens, compounds that mimic estrogens and can interact with estrogen receptors and ultimately like reproductive cycling and reproduction. But not only were the rhinos consuming phytoestrogens, but the microbes in the rhinos' guts could actually metabolize those phytoestrogens, and certain microbial communities could make them more potent, decreasing fertility even more, while other microbes could break down those compounds in a way that made them less potent. And so in order to understand this picture of infertility and ultimately fitness, it required looking at both the diet, host kind of processes, and microbial processes. And so that's an example of how the microbiome can affect host fitness. So now I'm just going to talk a little bit about environmental change itself. And so we know that natural environments are changing really rapidly as a result of anthropogenic activity. We're doing a lot of things to the planet that are changing habitats for animals in ways that require them to respond quite quickly. So they require novel phenotypes in order for the animal to persist. And, these can come about through several mechanisms. One could be in the short term within an animal's life through plasticity, reversible plasticity. And maybe in the kind of moderate term, you might see transgenerational effects like maternal effects affecting host phenotypes. And then, starting in the second generation and beyond, you have genetic adaptation, which can rescue animals from environmental change as well by providing different amounts of genetic variation in the population. And so these are three different processes that are probably very important for hosts responding to environmental change, especially when animals aren't able to migrate or disperse in response to that environmental change. But we really don't understand how the microbiome can affect these different processes. And so I'm going to talk a little bit about three different studies that I did, one during my master's and two during my PhD, projects that I've worked on to try to get at kind of a few aspects of trying to understand how the microbiome relates to fitness in the short term and in the long term. So the first question is, how stable is the gut microbiome longitudinally? Could it possibly serve as a mechanism of plasticity within an individual's lifetime? And so basically we don't really know a lot about how stable the gut microbiome is in different species. We don't know whether it changes on weekly, daily, monthly basis across the tree of life. And so I was really interested in characterizing those dynamics. The second is how gut microbiomes respond to environmental change across generations. So if one generation is exposed to some sort of environmental perturbation, how does that translate to changes in the next generation and beyond? And finally, how does this all kind of tie together to affect animal fitness in the wild, so in a natural setting? And so I'm going to start with the first question and tell you a little bit about some work that I did during my master's. So we know that gut microbiome composition is shaped by many factors. We've had a lot of different studies come out in in the last decade, starting to get at what type of factors affect the microbes that are present in an animal at a given time. And so that can be things that are more immediate, like the immediate environment, precipitation or temperature, or plants around an herbivore, or microbes that are in the soil. Those can all affect what microbes assemble in a host gut at a specific time. But also host phylogeny can also affect the microbes that assemble in a host at a given time. And so this is a figure showing the concept of phylosymbiosis, which is the idea that as host species have diversified, we see kind of in some species parallel divergence in the gut microbiome. And this has kind of produced a situation where oftentimes we see animals that cluster together, like all herbivores, having sort of similar gut microbial compositions, or the same species, like all Anolis lizards having relatively the same gut microbial compositions relative to other species. What we don't know is how those same factors affect the stability of the gut microbiome. And so thinking about this could start to inform us how different animals might use their gut microbiomes, or how gut microbiomes might function in different animals. So it may be that in some animals, the gut microbiome is incredibly stable from time point to time point, and in other animals, it's not. And so we sought to kind of test this using three different host species. So we started with rhinos, which were herbivores that eat plants. We had this really nice temporal sampling. It might not surprise you that they're herbivores that eat plants. That's pretty straightforward. We also looked at tigers, which are carnivores and unsurprisingly eat meat. But we also were interested in looking at this third species, the red panda, which is a carnivore but eats bamboo. And so this is kind of a historical decoupling between gastrointestinal anatomy and diet that we thought we might be able to use to look at maybe how these different factors affect gut microbial stability in different species across the tree of life. So what we did was we took these three species and we collected fecal samples in each of the 4 seasons. And these are the North American seasons. These are animals that are housed in captivity in North America. We sequenced their gut microbiomes, and 16S sequencing is basically the method that underlies all the studies in this talk. So just keep that in mind, and then analyze that data. And what we found is that we see really dramatic differences in turnover between different species. And so what turnover is using the Jacquard index. It's basically showing presence and absence between these two comparisons. So we have fall to winter, winter to spring, spring to summer. And so high values mean that there's lots of difference between the two communities. Lots of microbes have turned over or changed from one season to the next. And we see really, really high turnover in red pandas, really low turnover in tigers, and really kind of moderate turnover in rhinos. And so we see differences really dramatically between these three different species. We also see differences in synchronicity. And so what synchronicity is if you ordinate the microbial communities on like a PCOA, you can watch kind of in what directions the points move. And so this is just across one PC axis. You can see that all the rhinos, which are represented by these individual points here, move in the same direction at the transition between each season. So you see they change a lot between fall and winter, but they remain pretty stable, and all the individuals are responding to the same environmental changes in like the same predictable way. Versus in red pandas and tigers, their changes are not as predictable within an individual. Certain individuals are changing in one direction in one season, other individuals go down when some go up. And so we don't see as predictable of changes in those gut microbial communities. And so you can start to see how these communities seem to function quite differently for these different hosts. And so we're kind of interested in trying to generalize about what aspects of a gut microbial community might affect how stable it is or how predictable those changes are. And so we looked at both diversity and kind of connectivity in networks. And so the figure on the left here is showing just how many taxa on average are in the gut microbiome of these different individuals. And we see that rhinos have a really high diversity, while red pandas have this kind of moderate but very large variation in diversity, and this is on a log scale here, while tigers have a much lower diversity. But again, rhinos and tigers seem to have kind of narrower ranges of variability. And the same is true when we look at the connectivity. These are co-occurrence networks, so we look at what taxa kind of co-occur commonly, and we can also kind of look at how these links are positive or might be positive or negative interactions, like if the abundance of one taxa goes up and another follows it, or if the abundance of one taxa goes up and the other goes down. But what's kind of most interesting in this plot here is that red pandas have a lot of peripheral nodes, where these taxa don't really seem to be co-occurring or potentially interacting with any other taxa, while rhinos have much closer links, and tigers have, maybe less close links than rhinos, but still quite much closer than red pandas. And that's reflected in these degree distribution plots where really on the left here, this shows a lot of disconnected nodes in red pandas, where those lines are a lot flatter in tigers and rhinos, where they don't have as many zero links. So if you remember, red pandas are these herbivorous carnivores, which have different gastrointestinal anatomy than what their diet is. And so what we've kind of started to look at and hypothesize is that it might be an interaction between exerting control, or the ability to exert control over your gut microbes through some sort of long-standing interactions with them. Rhinos have these compartmentalized areas in their gut where they host specific gut microbial communities that are really important for fermenting their diet and breaking down plant material. And tigers tend to have a lower diversity gut microbiome. So those sort of factors, diversity, connectivity, and kind of evolutionary history, might work together to shape how stable the gut microbiome is in different species, and how it responds to environmental change, and therefore whether or not it could maybe serve as a mechanism of plasticity in some species. And so, yeah, just to conclude that section, basically we see lots of different flexibility in gut microbiomes. We don't really know exactly why that is. We have some ideas. And kind of the next steps for that would be to link some of those changes to actually functional, differences in host phenotype. So now I'm going to pivot to the second question here, which is how do gut microbes respond to environmental change across generations? So I just showed you that the gut microbiome can be very variable within one individual's lifetime. But what I was really interested in is if an individual is exposed to an environmental change, how do those environmental changes carry over into the next generations and throughout the second, third, fourth generation? And so we decided to look at this question by taking advantage of a conservation breeding program. And so this is a conservation breeding program for the Pacific pocket mouse, which is a species in Southern California that was thought to be extinct for over 20 years. They weren't seen. However, they were rediscovered in the 90s. And so there were really small, 3 populations that were rediscovered, very small numbers. And so a conservation breeding program was initiated at the San Diego Zoo in 2012 to try to increase these numbers. And they took a small number of individuals from the wild into captivity. So going from the wild into captivity is a dramatic environmental change. We've seen in a lot of studies that have shown kind of cross-sectionally that when you bring an animal from the wild into captivity, it can really alter their gut microbiome structure and function. But what was also really useful about this study context is that in conservation breeding programs, we keep track of lots of different metrics about host health and fitness. And we also keep track of pedigrees across generations. And so I can see how these changes in the gut microbiome, maybe they resolve immediately or whether they persist or what the dynamics of that are across generations. And so again, like I said, we know the gut microbiome can change within an individual, and that's exactly what we found. This is within the first generation. The time in human care is shown kind of in a gradient of colors. So the darkest colors are the later months in the first year after they were brought into captivity. And so we see individuals here are connected by these solid lines. And so we see movement generally towards the right of this PCA plot, showing that the composition is really changing in this direction across that first axis, which is the time in human care. So obviously it has an effect on their gut microbiome communities, but what about across generations? And so what we found was we looked at across all the generations. So we had F0 up through F4. And what we found is that in the first generation, F0s through F2, we see again this change across this first PC1 axis. Microbiomes are changing in their composition in this sort of direction, and that kind of movement persists through F1, F2. We see a lot of dispersion. The points are really far apart. Individuals are pretty different. And then after F2, we start seeing shift up PC2. And so these communities are still pretty dispersed. Individuals are pretty different before settling in F3 and 4 into a pretty new stable conformation. We don't really see the same thing in diversity. Basically what this is showing is that they're maintaining the same numbers or the same numbers of different taxa, the same level of alpha diversity or the different types of bacteria that are there, but the community composition is changing for multiple generations. And so because we're seeing the same level of different taxa, but different compositions, we decided to look at, what's going on kind of at the individual microbial species level. And so what we looked at was this is the proportion of different families across time. And so you can see here that the proportion, even it goes through this period of flux, but then kind of stabilizes off in F3 and F4. But underlying that are really kind of dramatic changes in the actual specific taxa that are found within that family. And so this Lachnospiraceae family is a good example where you see loss of this one species within Lachnospiraceae and kind of a new domination of the community by a novel taxa. And so the same thing kind of happens with Streptococcus. We see loss of this dominant Streptococcaceae member and replacement by a different strain. And so it seems like while the mice are maintaining bacteria in the same general families, they're replacing what specific taxa those are within that family. And basically what this results in is kind of an extirpation and replacement within that community, which creates this new captive community, which is quite stable in the end. And so what this plot is showing is we looked at what taxa were present in the wild mostly, which taxa were present in the captive across all the different mice, captive microbiome communities. And this light color here is the wild microbial taxa. You see that they drop off and kind of stabilize after generations 3 and 4. So they've lost most of their wild derived taxa. And this captive line here is this dark one. They've gained a lot more of these captive type microbes. So they have this new kind of stable community, but it looks a lot different than what they had in the wild. And when we look at some metrics that might be related to fitness, like weight, we tracked this across the same number of generations, but even into F5 and F6. And what this gray line here is the wild baseline. When we go and measure a bunch of mice in the wild, that's their kind of mean weight. What we see is that in the first few generations, we see a lot of flux. It's very different than what the wild mice look like. But after this 3rd and 4th generation, where they've kind of stabilized in their gut microbial communities, we also see them stabilizing in their weight a lot closer to what the wild mice look like. And the same is true for reproductive metrics. In the first couple generations after they're brought into captivity, estrus cycling is low, testes function is low. These are just 2 metrics that relate to reproductive readiness in these mice. But after generations 3 and 4, these metrics are a lot closer to those wild baseline. So the mice, after coming into captivity, it might take several generations for them to regain kind of stability in these fitness metrics and also stability in their gut microbial communities. And so what we kind of are hypothesizing might be going on here is the microbiome might be in some sort of healthy state to begin with in the wild, let's say. These mice are, adapted for the environment that they're living, let's say. Then this perturbation occurs where they're brought into human care. The environment is very different. The diet is very different. And that might shift the gut microbiome into some sort of unstable state. And so that's where we see lots of dispersion in the community, individuals very different. There's lots of change going on, and we see kind of parallel chaos in the reproductive metrics. And it might take several generations for them to shift into this new alternative microbiome state, which might also be just as stable and might also be just as functional, but might be made up of different microbial members than before. And so again, we need more evidence that's kind of mechanistic to try to look at what exactly is going on in terms of the relationship between those gut microbiome conformations and actual host phenotypes. So finally, again, I told you in the beginning that I'm interested in how anthropogenic change is affecting animal survival in the wild. And so this is kind of the focus of my dissertation work at the University of Nevada and at the Smithsonian in Panama, is trying to tie together how gut microbiome changes might actually affect fitness in the wild and kind of through what mechanism that happens, and whether or not these changes that we're seeing actually relate to host survival under environmental change. And so to answer this question, I'm working on the Panamanian slender ernal. So this is a little tropical forest lizard. They're small brown lizards that are found in lowland tropical rainforest. They're native there in central Panama. And they've evolved under the canopy of the rainforest, where temperatures are quite stable compared to other environments. And that has buffered them from any sort of dramatic temperature shifts. And that's produced a scenario where they have quite narrow ranges of thermal tolerance. Because of this, they're projected to be highly vulnerable to climate warming. They can't tolerate much more than a few degrees of warming before they start to really be effective physiologically. And so I've been working on this project, which uses an island transplant experiment to study the microbiome. So island transplant experiments have been really powerful, especially in evolutionary biology, to study the evolution of different traits in anolis lizards. And so we're doing something similar in the Panama Canal. So the Panama Canal is really interesting. You think of it as a place where it's very industrial, maybe lots of ships and concrete, but actually it's quite beautiful rainforest in a lot of areas. And so in the central region here, most of the Panama Canal is made up of Lake Gatun, which is basically just lots and lots and lots of flooded rainforest. And so that flooding of the rainforest to produce this canal has produced a scenario where there's lots of islands that used to be the tops of hills or the tops of mountains, and those have been kind of transformed into islands through flooding all of this, flooding all of this rainforest. So one of those famous islands is Barrow, Colorado Island. You might have heard of it. It's a kind of a tropical research hub that's right here. But there's also lots of little tiny islands. And so this is the size of like a boat that fits 4 people that we use to go in between these islands. And that kind of gives you a scale for how big these islands are. So we have a population of anolis lizards in the mainland. They're extremely abundant, these slender anoles. And so we're able to take small numbers of lizards from the mainland population, move them to these islands and start to look at environmental change. And that's really useful because these islands are quite different from the mainland, particularly in their thermal environment. And so kind of random changes in vegetation and canopy cover have reduced the overall canopy cover on these islands so that lots more sunlight gets in. They're a lot drier, they're a lot hotter. These are some plots showing you the differences in temperature between the mainland in green and the islands in blue. And so it's about 1.5 to 2 degrees warmer on the islands. The variation is a little bit larger. And so this is, again, showing you the breadth of the temperature variation and the means across all of our different island sites. And so we have 14 of those sites. And they're all relatively similar in their mean temperature. And again, 1.5 to 2 degrees might not seem like a lot of temperature variation, but for these guys, it really is very dramatic for them. And so what we do is we go to the mainland, we collect founding lizards, our founding population, we mark them and we measure physiology. So we measure their voluntary thermal maximum, panting threshold, which is another metric of upper thermal tolerance. We also measure metabolic rates. It's not included on this diagram, but we measure metabolic rates. We measure lots of different morphological traits. And we also collect fecal samples and tail tissue. And so the fecal samples are for microbiome analysis, and tail tissues are for building, for extracting host DNA and ultimately building pedigrees. These lizards, something I didn't tell you, they have a lifespan of about 9 months to a year. And so when we go back in the next year, we're able to catch their descendants. And so we can remeasure the same physiology, we can mark descendants, collect fecal samples and tail tissues again, and keep doing that for generations and generations. And so that way we're able to build these pedigrees and look at microbiome changes across generations and within a generation. And so this is a powerful system because on these islands, even though it's a closed system, they still have predators, they still have to compete, they still have to mate, they still have to find food. And so it's a lot more naturalistic, and it gives us an idea of how they might fare under climate change. So like I told you, the pedigrees are created from tail tip DNA. And this is some of the work that I've proposed to do during my Lawton, is working on creating these pedigrees and generating all of this microbiome data. And ultimately, these pedigrees and microbiome data will be used to estimate selection on microbiome traits and how that relates to selection on host phenotypes. And so these are pretty standard selection gradient methods. Landon Arnold pioneered these methods. Effectively, what you do is you regress fitness against your trait of interest and look at the slope. And that gives you an idea of how fitness is related to that slope and whether or not that trait is under selection. And so kind of the preliminary results, the data that I have so far is that I was able to do some sequencing showing that the mainland population, their gut microbiome has diverged already from the island populations have diverged already from the mainland populations. And they've kind of increased in their variation. And so this is on islands that have been established in 2017. And so these are islands that were the 5th generation of lizards when these samples were taken. And so we see that the mainland population is quite different than the island populations. But there's some overlap, but there's some unique composition here. And then when we look at why maybe those island populations are more dispersed or have more variation, we also see that there's clustering by island itself. So there might be some general selective pressure that's going on all islands in general that's causing that kind of overall shift in a certain direction. Maybe that's temperature, maybe that's precipitation or something else that all these islands have in common. But then we also see that islands kind of have their own microbial communities, and that maybe that's more related to the specifics of what insects are on that island, or maybe it's specifics of what the vegetation is like on that specific island. So there's both aspects here that we can look at. And so I also did this kind of longitudinal sampling of the mainland population shown in green here and these island populations. And so these are just temporally separated because we were sampling on the mainland for the whole season and we focused on sampling on the islands only later on. But what you can see is this is, again, one of those PC axes, and you can see that the mainland population is relatively stable over time, and the island population is relatively stable over time. So these are, again, islands that were established in 2017 and have been existing for quite some time. But what we also did in this particular year was we transplanted new islands. So we were able to get information about their microbiomes before transplantation and after transplantation and look at how they relate to kind of the mainland mean and the island mean, the old island mean, let's say, the ones that were established several years ago. So these are just the means for what the mainland was like over time and the island was like over time. And I'm gonna overlay on this the points that are for individuals that were transplanted in this particular year. And so on the left side of this dashed line, those are individuals that are pre-transplant or more similar to their mainland individuals, right? They haven't ever lived on an island. And then after this is within the same year where we went to go back and capture them within the same year. And what we see is that there's a transition really rapidly within a couple of months from this mean mainland phenotype to this island phenotype. And so basically what this is showing is similar to the mouse study. We see a lot of change within an individual's lifetime, shifting from kind of this mainland phenotype to an island phenotype. And so some of that is due to probably plasticity in the gut microbiome, changes in those compositions, because you can see that there's points that are lower than any that kind of ever existed before. And so obviously there have to be some changes in order for that to occur. But a lot of this variation is kind of within the realm of this mainland variation. It's just kind of more in the negative direction. And so one thing we wanted to do is looking at survival data with mark recapture, could we start to estimate selection on this aspect of microbiome composition, which is just one of the axes of diversity. And what we found is that at least on one of our islands and one of our populations, we found a significant selection gradient on this microbial composition variable, this principal component axis. And what that's telling us is that individuals that had microbiomes that were starting out kind of more in this negative direction were actually more likely to survive on islands. And so we don't know whether that has to do directly with how microbiomes might drive phenotypes or how phenotypes drive microbiomes. So for example, perhaps animals with a certain microbiome, those microbes are affecting thermal tolerance or some other aspect of physiology in a way that enhances their survival. Alternatively, individuals with a higher thermal tolerance or some other trait that helps them to survive on islands might just also be associated with having that particular microbiome composition. And so that's something that we've started to tease apart. And I've looked at that a little bit this summer, starting to try to manipulate gut microbiomes, changing their composition and seeing what the effects are on host physiology. And so seeing that microbiome composition, though, so far is correlated with host survival under environmental change, sets the stage to really investigate this further. And that's where the Lewontin Award will come in. And kind of what I've started doing is taking those F1 lizard fecal samples and those tail tips, extracting DNA and starting to build these pedigrees. So once we have these pedigrees, I'll be able to look at fitness not just through survival in the first generation, but also through reproductive success, which is a little bit easier for these guys because they're quite cryptic. And so mark recap is kind of a maybe not perfect metric of their survival. It's hard to find them sometimes. And so we'll really be able to look at how reproductive success and on these islands is related to gut microbiome composition. And so that's the work that I'll be doing with this award. And so with all of that, I just wanted to briefly mention that I think this research has implications for both basic science, but also conservation and management of species. In terms of basic science, we know little about the role that the microbiome plays in host response to environmental changes. Like I said, if it does turn out that animals with specific microbiome compositions have specific host phenotypes that are driven by those microbes that are under selection, then it's very interesting for the way that we predict host survival and adaptation under environmental change. And then in terms of conservation and management, both with the rhino example, with host fitness being affected by microbiome composition in captivity, and also with these conservation breeding programs with these mice, might tell us that it takes a few generations before individuals reach a new stable microbiome composition that might relate to stabilization in host physiology. So there's a lot of work to be done in terms of the directionality of these correlations and the mechanism kind of underlying this. And I hope to kind of get at that with the additional work I'm doing in my PhD. But with that, I'd like to thank, of course, the Societies for the Study of Evolution for this award. That's really enabling me to kind of do the rest of my dissertation work. But also all the people that have helped with these different studies over the years. The island transplant study in and of itself is a team of 10 people every year. So the list of acknowledgements is growing and growing. But all the people at the Smithsonian who support, and facilitate this work, and at the San Diego Zoo as well, have really made all of this possible. So with that, I will finish up and take questions. I don't have a question slide, so I'll just go back to this one.
Thank you, Claire. Thank you very much. Very cool talk, a lot of results, and I really like the drawings as well.
Thank you.
Yeah, very nice. So I'm happy to take questions either from the chat or from the room, which has actually grown quite a bit since we started the seminar, so that's nice. Yes, Howard.
Hi, I just want to echo Anne's comments. Those are beautiful slides. It's really well designed. I was curious, apologies if I missed it because I came in a minute late. I'm curious if the high variability in gut microbiome among red panda individuals could be related to the sampling protocol. How were individuals collected, and could you have hit upon more unique populations in that species compared to others that could, if there's among population difference, for instance?
Yeah, so that's a great question. This paper actually it just got accepted, and one of the reviewers also asked the same thing, like, are there differences?
Wasn't me.
Yeah, I know who you are now. So differences in sampling or anything. And so we went through a lot of effort to try to look at, are there certain variables that structure these differences that are other than host species? And nothing that we looked at. We sampled at 2 different zoos, and we found the same results at the 2 different zoos. we looked in males and females, and the same thing seemed to be going on. And so this is actually like, it wasn't a very surprising result, because there actually are other studies in giant pandas that showed the same sort of really dramatic high level of variability in giant pandas. And so, yeah, kind of the working hypothesis is that pandas are unique because they eat this herbivorous diet, but they don't have any sort of anatomical adaptations to facilitate that. And so eating plants might be introducing a lot of different microbes, either from the soil or from the plants themselves, that are different among these different plants and among these different individuals. But a lot of them are highly transient taxa that are just kind of moving through, because red pandas have a really rapid digestive passage
Apparently,
time.
maybe I misunderstood then, because I assumed that part of the idea was that red pandas have a highly variable diet because they ate both meat and plant matter, but that's not the case. They're plant only.
They eat 99% bamboo.
Wow. Okay.
Yeah.
That's interesting.
Yeah.
Thanks.
So actually, can I rebound on this? Because if I understand well, in this study, all the animals, so the tigers, the red panda, and the rhinos, they were all captive, right?
Yeah. Sorry, that's another, I got a little sidetracked there, but yeah, the sampling was done. They were all captive populations, and it's done in very similar ways in terms of the freshest fecal sample we could find, and we took multiple from different individuals and kind of averaged the results. So yeah.
So I was wondering if there are any data, if there is any data in the literature, for instance, for red panda, since it's puzzling Howard, whether you can check whether these captive populations are representative of what you would find in the wild. And this is particularly because you showed in your chapter 2, comparing captive and wild populations, that it can differ a lot. So I don't know if the predictability results, I thought it was very exciting. I don't know if it's something that is known or whether it has only been tested in this kind of captive situation.
Yeah, I think to answer that, I think it's really only been tested in captive situations. There's not a lot of literature about gut microbiome stability in the wild. And the other thing about that study was that we sampled at the individual level. So we tracked the same individuals over time, and that's really challenging in the wild setting to get kind of that consistent sampling. So I don't know that there's a lot of evidence as to how this functions in the wild. So it's definitely an open question for sure.
Okay. We have a question in the chat on a different part of your talk. Jasper says, great talk. Sorry if I missed this, but are the transplant microbiome results based on abundance or presence/absence?
Those are based on abundance. It's a metric that takes into account abundance and presence, absence.
Okay. I have another question, and it's more related to what you said would be your next steps. So you're gathering the data to construct your pedigrees. And you mentioned that you will use these pedigrees to measure selection, because then you can have a better fitness estimates. Actually, I'd be keen to know how you do capture recapture, but this is another question anyways. But I'm wondering if you or someone else in the group has the objective of using this pedigree not to look at selection, but to look at quantitative genetics, following the equation that you showed early on in your talk. So it would be really cool to decompose the different variance components and see whether variance attributed to the microbiome, if it's not taken into account, whether it kind of pollutes the genetic variance or the environmental variance.
Yeah, I think that's a really great idea. It's not an experiment that I explicitly have planned, but I think it's a really useful thing. I think that particularly using like maybe a common garden sort of approach, like taking individuals that have been diverging on these different islands and bringing them back into a common environment, I think could be a really powerful way to look at that along with the pedigree data.
Yeah, and I guess there aren't that many estimates of heritability of microbial communities, so that could be
Yeah,
very
yeah.
cool,
That's
depending on your.
another oh, sorry.
Yeah.
Keep
No,
going.
no, I was going to say depending on how connected your pedigree is and your sample size, obviously. But
Yeah, that's another kind of big part of my dissertation. The other part of this that I didn't talk about is the heritability aspect. Because especially in lizards, these are oviparous lizards. We don't know how heritable these communities are. So even if there's selection occurring on the microbiome, we don't know whether or not that translates to like some sort of host response, if that's possible, because we don't know if they're heritable. And so that's kind of the other side of the coin, is estimating heritability, but it is challenging because yeah, for heritability in the microbiome, we need, multiple samples to try to get some sort of estimate of stability, which we have that in some individuals. And then once we kind of can characterize that individual's microbiome, looking at that related to the pedigree. So it's something that we hope to do if we can get enough, like sampling events for each individual.
that sounds very cool. Any other question? Well, maybe I'll have a last question. Sorry, I have a lot of questions, but related to what we just discussed. So this is a very general question because I don't know enough about microbiomes, but you probably have a better idea. So I was mentioning that initially in your talk, you gave this equation of like variance components. And so I'm wondering whether we actually know exactly whether the variance in microbiome would be confused as VA or VE, but also you also talked about the mechanisms, so plasticity and evolution. And are there enough studies now that we know whether microbiome communities, they trigger more plastic response, or whether they more loop in the evolutionary response? This is a very general question. I'm just wondering where the field is there.
Yeah, no, it's a great question. I actually just read a preprint of like kind of a meta-analysis yesterday that was looking at the role of the gut microbiome in like host plasticity, and effectively the conclusion of their study was that we don't have enough studies that are mechanistic, because so basically a lot of this work has been done with just looking at the 16S microbial composition of like the taxonomic composition, and that's really doesn't give us any information about community function, and oftentimes these studies aren't really well paired with measures of host physiology that actually show how these things relate. So from their paper, to answer your question, I think we're very, very early on in understanding whether or not this actually functions this way or doesn't. In terms of the first part of your question about microbiomes and environmental variation and additive genetic variants, I think like that's another thing that I've thought a lot about, because I think before we would have just kind of lumped all of that variation into the VE component, right? Because if it's not part of genetics, then it must just be part of the environment. And so I think that's, the question is, if we do account for some of that microbiome composition or function or some sort of trait, does that reduce the VE, because we can explain some of that via the microbiome? And so I think that that's still a very open question.
Yeah.
Oops.
It would be very cool if you had an example of this with your data. So there's another question from Jasper online. I'm happy for Jasper to unmute, or I can just read the question. So the question is, for your question 2 or 3, have you looked into whether the changes in microbiome composition is mirrored by changes in functions within the microbiome? Are there any specific functions that you have seen or would predict to change?
Yeah, that's a good question. So one thing that I would really like to do is use a different approach, not 16S, but use either RNA-seq or shotgun metagenomics to look at bacterial genes and what genes are being expressed. because yeah, the 16S data doesn't really give us good information about function. There are some tools where you can try to map back the 16S gene to like what bacterial taxa that is and what functions might be encoded, but it's not really good in species that aren't well studied. So we don't really have a great idea of what these bacteria are doing, but in terms of what functions I might predict to change, not that I've seen in my specific lizards, but what's been shown in some other species is that microbes can produce heat shock proteins that can mediate thermal tolerance. And so that would be maybe one thing that might be changing in an environment that's hotter and drier are maybe the microbes and how they affect thermal tolerance in that way.
Great, thank you. So if there's no further question, I want to thank you, Claire, for taking the time to prepare and present this beautiful presentation and answering our questions. We still have 2 GREG seminars left, maybe a third one. I don't know if the one that was canceled is postponed or not. But anyways, we'll see, have news about this on the website. And I wish you all a very good day. Thank you for coming.
Yeah, thanks so much. Great talk. Really appreciate it. All right, thanks everybody.
Bye-bye.